decided I'd use my computer because I didn't want to be a geek patrol guy. So, and also Dr. Melling's video didn't work, and I think it has to do with the software. All right, my name's Louis Clarke. I'm a rehab doctor in Clear Lake, Texas, which is right between Houston and Galveston. I've done a lot of work previously on neuroscience and did a PhD in that and at Southwestern in Dallas. My interest has always been neuroregeneration and neurorecovery. In the 90s, it became clear that we can generate new brain cells. We can actually make new neurons. There had been a lot of resistance up to that point, and it finally became clear that there was some neurogenesis from stem cells in the CNS. So I was fairly disenfranchised with my current therapies. When you put a stroke patient in the hospital or spinal cord patient in the hospital, it's rare that they don't return with countless complications, especially in the stroke patients. They usually have another stroke within a few years. So I felt like we weren't doing anything. We had no disease-modifying strategy. And all of the strategies that have been presented, statin drugs and the countless pharmaceuticals, absolutely do not appear to have a dramatic effect on stroke prevention down the road. Nor do they really contribute to stroke recovery or recovery of function. So I went back to my neurochemistry, biochemistry roots and decided, what can we do to generate neurogenesis? How do we redirect our own stem cells into becoming neurons, glia, or oligodendrocytes? So that's what we've been working on for the past 20 years. So it's very difficult to do anything with drugs in the United States because of the limitations. So we fell back on just basic biochemistry. What mechanisms are involved in doing that? So over the years, we have isolated all of the neurochemistry studies, all the neuroscience studies that we can find that seem to contribute to neurogenesis. And we just put them all together. And I know that sounds like a shotgun effect, but it's very effective. So at the end, I want to show you a video of a lady. But before we do that, I want to talk about what goes on at the initial stage of a stroke. And that's where we seem to make our biggest mistakes. When a patient shows up in the ER with numbness or tingling on their face or a little slurred speech, they do a CAT scan. If there's not a bleed, then they just pretty much go on and say, well, I don't see, you might need to see your neurologist next week. But by the time they get home, 2 hours later, their arm is numb and not moving, and pretty soon they're hemiplegic. This happens over the course of the first few hours. So what's going on in that first few hours that we can mitigate by early intervention? Initially, in the first hour of the injury of neurons and glia, you have this huge release of free radicals. The second phase is that you have the neurotoxic effects of the adjacent neuron, on the adjacent neurons. So the neurotoxins like glutamate, aspartate are released from the damaged glia. They're released from the neurons that are killed off with the initial phase of the stroke. So now these neurotoxins, these excitotoxins, bind to the receptors on the healthy neurons, then killing those off. So now you have a secondary wave of neuronal damage. Then the inflammatory cells move in and try to clean things up. And those are cleanups that you don't want necessarily, because now they're going to clean up cells that are healthy also. So all of the interleukins and all of the macrophages and everything that is the sequela of inflammation, you want to try to rein in. So if you address those in the ER, you have a much higher survival rate, which with a dramatic decrease in neurologic deficits. So the deficits that we've seen seem to establish themselves in the first 24 hours after stroke. And if we intervene then, we win. So what all can we do to fix that? We know that even in the microtrauma, I'm working with a lot of the NFL players now. They have cumulative traumas, and there is a lot of talk about CTE. And we know now that within the first hour, you already have a laying down of the protein carbonyls, which are the oxidized forms of proteins, intracellular proteins and extracellular proteins. These are the proteins that make up beta amyloid. You also have, in this just minor damage, you have a release of these phosphates, and you get this hyperphosphorylation of the tau proteins. So already within the first hour, you're having this progression toward degenerative changes in the nervous system. And when you're young, you have lots of reserves so you can bounce back. But what happens over the decades to come? And we know that there is an increased incidence of all of these things: Alzheimer's, ALS, multiple sclerosis, Parkinson's disease. And there's a lot of debate on whether or not there's something you can do to mitigate that early on in players, in children who are in contact sports. So this is just a representation of the tau proteins, how they hold the axon together. Let me get this thing. Nice thing about giving a lecture early is that the laser still works. So these tau proteins hold these microtubules together, and as soon as they get hyperphosphorylated, they dissociate, and now you have this axon that just frays like an old rope. And eventually you get neuronal death, which looks very much like this. This neuron just turns into a dying bag of nothing. And if you combine that with the concurrent development of the beta amyloid, you end up getting all this goo that plugs up this neurotransmission. So what we decided, can we make an intracranial helmet? Can we prevent this? Because obviously the helmets on the outside don't do much good. So what we decided was to put everything together that we can find that will protect neurons. And it turns out that the same things that protect neurons also generate neurons, and we'll go into that as we go. The first stage, as I talked about, is to scavenge these free radicals. Got to do that up front, block the release and receptors for the neurotoxins, and decrease the pro-inflammatory cytokines. And if we can suppress the microglia and the macrophages, the gobblers, we might actually be able to intervene. This is a substance called PQQ. And the first set of slides, I just want to talk about what we can use to scavenge free radicals. And this goes with everything. You can use it for any injury. And again, everything is predicated on can you grow seeds, tomatoes in sand? And the answer is no, you cannot do that. You have to fertilize the garden. So in this case, what we're doing was we're throwing in all of these chemicals that we know mitigate this damage. So pyrroloquinoline quinone, or PQQ, it suppresses reactive nitrogen species, it blocks inducible nitric oxide synthetase, and we know that it does at any ischemic event. It also, so there'll be more to PQQ as we go. Vitamin K2 is a very little known vitamin K. Unfortunately, we just don't talk about it. We were so worried about complicating Coumadin therapies for AFibs and other things that we try to stay away from vitamin K. And yet, vitamin K does all of this blocking of the free radicals. But you'll also notice, as we talk about later, vitamin K2, which has very little effect on coagulation, also works like coenzyme Q10 in electron transport at the mitochondria. So we're now beginning to see that maybe vitamin K2 and all the quinones for that matter might improve mitochondrial function through similar functions as CoQ10. Vitamin D inhibits the metalloproteinase of the iron and the zinc oxidation of membranes. And then the neurosteroids, DHEA, pregnenolone, progesterone, these are all generated de novo in the brain. These hormones have, and we'll talk about them when we talk about neurogenesis in a minute, these hormones have amazing potentials to decrease inflammation and also oxidation. Acetylcarnitine, on this slide we're really talking about mitochondrial protection. And the acetylcarnitine, it prevents the electron leakage from the mitochondria. CoQ10, all its redox functions with its electron transport. It also regenerates vitamin E and C and their oxidative potential. Alpha-lipoic acid is a spin-off of the Krebs cycle, and it actually is there right in the inner membrane of the mitochondria, and it scavenges. And it has a lipophilic end of the molecule and a hydrophilic end on the other side, and so it can go into any tissues, which is very handy. So it can regenerate both E and C as well. Melatonin is probably the most important thing in the brain as far as healing and protecting. It has direct free radical scavenging capability, but it also stimulates these antioxidative enzymes. It reduces the electron leakage from membranes and also augments the other antioxidant efficiency. So the second phase is to try to suppress the neurotoxin release or the receptor binding capacity of these excitotoxins. And look, with same stuff, the same things do that as scavenge the free radicals. Now PQQ decreases the N-methyl D-aspartate receptor, which is really seems like the villain in everybody's judgment as far as excitotoxicity, released by the adjacent glial cells that are damaged, bind to the neurons, kill them off. So PQQ can decrease that receptor affinity through some kind of maybe an antioxidant redox thing. Nobody really is quite clear on what PQQ does to protect neurons and decrease this receptor affinity for NMDA. Vitamin K2, again, specifically seems to protect oligodendrocytes, although it does have some benefit with neurons from this glutamate-induced cell death. Glutamate and aspartate bind to this NMDA receptor, and that's what kills off the cells. Vitamin D, that reduces this NMDA receptor sensitivity by decreasing the calcium influx so that the receptor doesn't work as well. So you're desensitizing at the receptor level. Here we go with the neurosteroids again. It's the only neurosteroids that actually has been shown to decrease NMDA receptor sensitivity. Back to the mitochondria, acetylcarnitine protects these neurotoxins. These are exogenous neurotoxins, but it's clear that they also protect endogenous neurotoxins as well. The B vitamins, all of them reduce NMDA excitotoxicity. And melatonin, as we talked about, is extremely important in the CNS, and it's a potent NMD receptor antagonist. Alpha-lipoic acid, same thing. Ubiquinone, same thing. Inhibits NMDA receptor-induced cell death. Straight out of literature. The third thing we want to do is suppress the pro-inflammatories. We've got PQQ again, lo and behold, suppresses interleukin-6, decreases C-reactive protein. Menaquinone, vitamin K2, suppresses interleukin-6 and the genes involved in the acute inflammatory response. Vitamin D increases interleukin-10 by 43% in the vitro studies, decreases interleukin-6, and tumor necrosis factor alpha activates the MKP1 and interrupts the LPS inflammation cascade. DHA, pregnenolone, and progesterone, back to the neurosteroids. This activates the CD55 protein after a contusion, which inhibits the complement convertase that would activate the inflammatory cascade. So each one of these things is shutting down the inflammation response to some degree. Melatonin is an interesting tumor necrosis factor alpha blocker. And I use that in cancer patients that get quite cachectic. And I can give them up to 30 milligrams. Russ Ryder, another neuropharmacologist, done all the research on melatonin, known worldwide as the melatonin man. A lot of fun to hear his lectures. So I give these patients 30 milligrams at bedtime, and they begin to eat. We don't use Megace. We don't use any of those glucocorticoids. We use melatonin. And not only that, they sleep quite well. The B vitamins, they're anti-inflammatories both with and without the methylation of homocysteine, which is how it converts homocysteine back to methionine, which is the building block protein. Independent of that mechanism, it still is an anti-inflammatory. We know that osteocarnitine prevents inflammation in the kidney, and we're assuming that it does in the brain. Alpha-lipoic acid, again, back to the metalloproteinase, these proteases that metalloproteinase 1 and 9, they are all equally inhibited by both CoQ10 and alpha-lipoic acid. So here we're intervening. We're saving the mitochondria, save the world, save the cheerleader, save the world. We're scavenging the free radicals, and we're increasing antioxidant gene expression. We're blocking the release of receptors for neurotoxins, and we're decreasing pro-inflammatory cytokines. So the second thing we want to do is we want to start promoting neurogenesis. So the same things that are mitigating damage are also promoting neurogenesis. We'll talk about bioactive phospholipids in a minute, but we'll first of all get to the things that we were already using. We're increasing mitochondrial biogenesis with PQQ, also increases neurogrowth factor release from the astrocytes. Vitamin K2, that menaquinone, mitochondrial electron transport, I alluded to that, similar to CoQ10, fascinating. We might actually be learning more and more about all of the quinones in electron transport in the mitochondria before too long. CoQ10, we already know about electron transport, and OXPHOS, it also generates more mitochondria. The most important mitogenesis, mitochondrial biogenesis, is really this PQQ, a fascinating molecule, and probably Dr. Davis will mention that later in some of his interaction. Osteocarnitine, we know how that, of course, that's how we burn fat in the mitochondria with beta oxidation. Osteocarnitine, great at crossing the blood-brain barrier. So the brain cells can function for a short period of time without any oxygen and glucose, and salvaging the neurons with high doses of osteocarnitine has been attempted, and there is some positive outcomes on that, but I don't have those data here. Alpha-lipoic acid, again, mitochondrial biogenesis. I'm making new mitochondria through a totally different pathway. Vitamin D increases the mRNAs, which encode proteins that increase mitochondrial biogenesis. And melatonin, our old friend, continues to generate new mitochondria. But interesting in this whole thing is the contribution of the neurosteroids. Why would there be neurosteroid generated de novo in the brain? And I am certain that it has to do with redirecting stem cells into neurons or glia or oligodendrocytes. This sigma-1 receptor is a receptor that is the interface between the endoplasmic reticulum and the mitochondria. They actually touch right there. And the cell-cell communication between those is due to phosphatidylserine, all the phospholipids. And the sigma-1 receptor acts right there, and it generates neurosteroids with stimulation. The neurosteroids then feed back and generate more neurosteroids. And what happens then is you get a neuroprotection, but also this neurite outgrowth. And You see how it's integrated with the phosphatidylserines, ethanolamine, and choline, and all of these neurosteroids are sigma-1 agonists, and they all do the same thing. We already talked about DHEA blocking the NMDA receptor for neurotoxicity. We know from the literature it prevents the extensions of CVA and spinal cord injury, and it inhibits all these inflammatory chemicals. We also know from other studies that it decreases neuron death and ischemia reperfusion, maybe by blocking some of those neurotoxic chemicals. We already talked about it binds sigma-1s, increases neuronal survival, stimulates neurite growth, and directs stem cells specifically in the hippocampus. Now that's fascinating, because when I start these patients on this whole cocktail, an Alzheimer's patient, within 2 weeks, they become more lucid, they begin to recognize their children that they hadn't known in 5 years, and it is just fascinating to watch. Allopregnanolone promotes neurogenesis and oligodendrogenesis and white matter formation. It decreases neuroinflammation of beta amyloid in Alzheimer's. It's a ligand for the sigma-1, as we talked about, with neurite formation, and it regulates repair and regeneration. Progesterone is not just for girls anymore. It directs stem cells toward oligodendrocytes, so I use a lot of progesterone in multiple sclerosis and in all of the demyelinating diseases. And that, together with vitamin D, I have taken them into remission. And I know it has to do with just those two specific things. So if men can take progesterone with impunity, it's a great hormone for all of us. It also happens to shrink their prostate, which is usually handy. But some of you guys are too young for that. It's negatively correlated with beta amyloid and tau in some of the brain regions, and it's neuroprotective. In the ALS mouse model, it normalizes mitochondrial complex 1 activity in the cervical cord motor units. So the next slide, I want to show you this little video. It's a video of a lady who's in her 80s, and she had a bleed in her cervical cord, high cervical cord. It rendered her Brown-Séquard cord. Brown-Séquard cord is a syndrome where you have loss of sensation on one side, loss of motor on the other. So you're completely paralyzed on the right side, couldn't feel anything on the left side. So when I got her in the rehab hospital, she was already a couple of months into this, and she had had no recovery. She's still spinal cord injured, essentially. So this is what happens when we started the cocktail. And this is where hopefully I can get this to play. Can you hear that?
When you got here,
All right, we do a lot of cheerleading, as you can tell.
oh, that's marvelous.
I'm so proud of you.
That is really amazing.
All the way. Right. Come on, say praise Jesus. Come on.
Cheap for that left hand. Can't praise. Gotta praise.
She's just now beginning to get the distal motor back. She's starting to move her fingers and hands at this stage.
Kick that leg out like a can. Look at that. Oh,
Beautiful.
that's beautiful.
That is amazing.
So what we've had a follow-up since then, and now we've got her walking greater than 300 feet. She uses a rolling walker. She's even able to advance that right leg. She's feeding herself with her right hand again. There's a dramatic neurologic recovery when long after you would expect spinal shock to have recovered. So that's what we're doing with this little combination of things. The nice thing about what we did with all of these chemicals, and there's quite a few more that we added in there that we didn't talk about today, is that we put them in a powder packet so I can get them down feeding tubes. I can use them in the acute setting in the ICU. Works very nicely for neuroprotection and neurogenesis. There is a latency that it seems to be about 2 weeks when we start this protocol, when they seem to begin to recover function. And I'm pretty sure that has to do with some kind of stem cell migration and or differentiation. I don't have any evidence for any of that, except that seems to be the time frame for some of that in the animal studies. You guys would probably know a lot more about that than I. But thank you for listening. It's an interesting function.
We have time probably for one question. Go ahead.
I'm just curious, with your cocktail that you administered, did you do any CTs or MRIs pre-post?
No, I haven't. I haven't done any of the follow-up. Neither with the MS either. I'd like to see if some of those plaques regress.
Is this regulated? I mean, is it that a lot of drugs? You know, how do you do this?
Well, nothing's regulated. Everything is a supplement.
Supplement. Okay.
So I'm out from under that umbrella, which is nice.
Right.
And it's easily accessible by all the parties.
Great. Well, thank you very much.
Very
Oh,
welcome.
so